Hi, I'm Nika, and today I will introduce GenStore. Genome sequence analysis is critical for many applications. Genome sequencing machines extract the smaller fragments of the original DNA sequence known as reads. Read mapping is the first key step in genome sequence analysis that aligns reads to potential matching locations within the reference genome. Read mapping requires computationally expensive approximate string matching ASM to account for differences between reads and the reference genome. Read mapping performs ASM on large genomic data sets containing millions of reads. Therefore, read mapping is both computationally expensive and incurs high data movement overhead. There has been significant effort into improving read mapping performance through efficient heuristics, hardware accelerators, and various filters that prune reads that do not require expensive computation. While these approaches address the computation overhead in read mapping, none of them alleviate the data movement overhead from storage, whose impact becomes even larger when computation overhead gets alleviated. Our key idea is to filter reads that do not require the expensive computation in the storage system to fundamentally reduce the data movement overhead of read mapping. However, filtering reads in a modern SSD can be challenging due to different behavior across read mapping workloads and the limited hardware resources in the SSD. By addressing these challenges, we propose GenStore, the first in-storage processing system designed for genome sequence analysis to reduce both the computation and the data movement overhead. GenStore provides high performance and energy benefits compared to the state-of-the-art hardware and software baselines. For more details on GenStore, please attend our talk on March 3rd, session 6A.